All right, hello everyone, and welcome to the little review video here for Physiology 2, Exam 3. So I did a little differently this time where I just wrote it all out in bullet point form, so there's a lot here, but it's less I have to write then, and I'll just talk through as I go through this and draw anything I need to draw. Now, I'm not sending out like a Word file with this bullet point list on it, so just watch this video and see what you're missing and have that help you figure it out. Hold on, I'm going to adjust the little captions here quick. So they're not chopping off at the top. There we go. Okay, so the captions should be good down there now. Okay, so let's go through it. Remember, this is on chapters 20, 21, and 23. Chapter 20 here is the lymphatic system. Chapter 21 is the immune, and chapter 23 is the digestive system. So not as many chapters as previous exams, but still requires a good amount of studying. Similar format to previous exams. I graded out at 85 points, but there are 90 points total. True, false, matching, multiple choice. Practical is mostly based on the digestive system, just as a heads up, because it's based on our classroom models. All right, so going through lymphatic system first here. First off, we have our lymph vessels, and know that they are more like veins than arteries because they have the valves and they work by skeletal smooth muscle pumps moving the fluid through. So remember, they bring that fluid in at the capillaries, and then it moves through the system. Fats are slightly different, and this kind of relates to digestive system as well. So remember, these fats are delivered to the body via those lacteals. I'm going to change the color here. Let's go to a bright red. There we go. Lacteals in the lymphatic system. Oh, this is acting weird. So lacteals in the lymphatic system. Remember, we have the little villus, and in the middle of that villus, we have that lacteal coming up. So remember, this is a villus. This is a lacteal in the middle. And you have all these cells on here that are ciliated in the small intestines. But fats are absorbed in these lacteals here. And then this kind of goes into the immune system as well, but B cells mature in the red bone marrow, but T cells, they're made in the red bone marrow, but they have to go to the thymus to mature, so slightly different. Both of them can then go out and go through the circulation. They can go into lymph nodes and so forth. And then understand the importances of the lymph nodes. So, lymph nodes are secondary lymphatic organs, and as fluids are moving through them, so if I were to draw a lymph node here, let's give myself a little bit more space. So let's move this down to here like this. So if I were to draw all these afferent pathways coming into a lymph node, and then we have a little, lymph node here, there might only be one efferent pathway going away. So then here would be the structure of the lymph node. You have all these little trabeculae coming down here, these germinal centers in the medullas, and the medullary cords coming in right here. So these are all inputs coming in, so more coming in, less going out. This allows the fluid to slow down substantially, spend more time in here. So slow down the movement of lymph and are considered small secondary lymph organs. So the structure of the node, so I went over it a little bit here, but make sure you understand the anatomy of a lymph node and then how these more afferent than efferent slows the fluid down. And then also in these little germinal centers here are where B cells are seated and sitting and waiting. There's typically sitting on reticular fibers sitting in there. Okay, then you understand the main organs of the lymph system and the vessels discussed in that lab video. So I made that lymphatic system lab video showing the general features of the immune system, not the immune, the lymphatic system, and how you have the thoracic duct, that cisterna chyli, and things like that. So make sure you review that. So some of the organs here, just to mention, tonsils are the ones found in the oral cavity. So you have a couple different tonsils. You don't have to know the different ones, just that's where tonsils are found. Peyer's patches, just jumping ahead here, are found in the distal portion of the small intestine. So why would these Peyer's patches be found in the distal portion? So these are also MALT, or mucosa-associated lymphoid tissue. So remember, in the large intestine, you have a high amount of bacteria. That's where all the bacteria live. So some of those can come back through that ileocecal valve. So it makes sense to have a higher amount of lymphoid tissue in the distal end of the small intestine to help protect against any bacteria that might be coming down through. And then remember, as we age, the thymus is more active when we're younger, and as we age, it doesn't work as well. So that's, lymphatic system was a short chapter, but make sure you watch that review video and that lecture, of course, and check out the virtual labs and the Kahoots that go with that could help you answer some questions. Then the immune system here, This is the more heavier chapter of this unit because it's a lot of new concepts, a lot of new cells, and so forth. First, understanding the inflammatory process, so pain, redness, heat, and swelling, and the importance of all those. So that heat, the purpose of having a fever, is beneficial because it activates immune cells, and heat in that local area allows your immune cells to work faster and also sometimes could prevent bacteria from replicating. So fever isn't always a bad thing unless it's too high. And then the whole inflammatory process also allows more leukocytes, particularly neutrophils, to come to the site of infection and begin fighting. And then understand the different lines of defense. So like, we have our first lines of defense, our second lines of defense, and our third. So first and second are part of the innate immune system. And then third is the adaptive. So, one of the first lines of defense are, the mucosa linings, our skin, and so forth. Second would then be things like our natural killer cells, our macrophages, dendritic cells, things like that. Then adaptive, this one involves B cells and T cells. So B cells, T cells, you have to go activate them outside of that area of infection. So then with the innate, we also have interferons. So understand how interferons protect neighboring cells from viral infections. So one cell gets infected by a virus, it produces a signal, which is the interferon, which goes to the neighboring cells and says, hey, look, I'm going to die. Here's how you can prevent that virus from replicating inside you. So that could allow the virus to not bind to a receptor, could prevent the virus from replicating inside. Somehow there are different sorts of interferons, but they could protect against the cell in different ways. And then here, like I mentioned before, understand the difference between innate versus adaptive. So innate does not involve B and T cells as part of the first and second line of defense. Adaptive adapts, and you can typically form a memory response from that adaptive, so it's more specific. And then with vaccines, understand how vaccines work. So vaccines work by priming the immune system, and that way if you get a secondary exposure, you have those memory cells of the adaptive system there waiting to work. And then with adaptive immunity, just make sure you understand the difference between passive and active immunity. Active immunity forms memory. Passive immunity is just temporary and you don't form memory cells. So this could be acquired naturally or artificially. Natural passive immunity would be something like breastfeeding, or acquired would be giving the antibodies to attack a certain things like we've done with Ebola. So here the key is passive does not produce memory cells. And then understand the difference between cellular. So for breaking down adaptive immunity now, understand the difference between cellular immunity and humoral. So humoral involves B cells, cellular involves T cells. So B cells are releasing antibodies. Antibodies, remember, are produced by plasma cells. So this makes them humor. So humor is a fluid. So these move through the fluid, whereas T cells are specifically attacking cells. So then a little bit about antibodies. Remember here we have the general feature of general structure of an antibody. We have our heavy chains and our light chains and our little disulfide bonds that hold them together. But remember, these are proteins. Some people confuse antibodies and think they're cells. Antibodies also do not kill. They can inactivate, they can agglutinate, they can label them for destruction, but they do not specifically kill that pathogen. But then here, these are the main warriors of the immune system. So T cells and B cells are the main warriors of the immune system. And then understand how cytotoxic T cells kill a cell. So here, if we were to draw two cells in here, so this is the infected cell, and then this is the T cell over here. So here, T cell, this is the killer T cell. So what it does is it releases vesicles here that begin puncturing it and forming pores. Porphyrins form pores. So these porphyrins form little holes in the cell membrane. Then other vesicles then come out and release these granzymes, which then induce apoptosis of the cell and the death. So porphyrins form pores, and then granzymes go in and induce apoptosis. And then here, just as a reminder of some different cells we find in the immune system. Antigen-presenting cells, or APCs. These can be dendritic cells, macrophages, or B cells. Dendritic cells here are the main ones. So they are the initial responders to the immune system. So let's say a pathogen gets in your skin. Dendritic cell eats it up, presents the fragments on its surface, runs to the adaptive immune system and says, hey, look what I found, mount an immune response against this. And then also, we can talk about clonal selection here, specifically of T cells. Remember, there's positive and negative. Oop, that's not a negative sign. Positive and, negative clonal selection. Positive clonal selection makes sure those MHCs, or major histocompatibility complexes, match. The negative selection makes sure the antigens don't match something in the body. If they match something in the body, they could be released and cause an autoimmune disease. These are typically regulated by regulatory T cells, which I'll talk about down here. So regulatory T cells then prevent this from happening, but if this is then released, it's then activated and then can go throughout the body. But clonal selection ensures T cells do not match a body's own cells, especially at that antigenic spot. It wants to match the MHC. Activated T cells and macrophages release the little immune boosters, or Red Bulls of the immune system, called cytokines, and they mobilize other immune cells and leukocytes to the area of infection or wherever the immune response is happening. And then also, we can form this membrane attack complex via the complement system. And so if we have a cell membrane here, let's increase some space. So if we have a cell membrane here, phospholipid bilayer, this membrane attack complex comes in and forms a large pore in it. This allows anything to come in and out. So it forms a bunch of these pores in, and that cell is no longer able to regulate permeability. So osmosis is no longer regulated, and what happens is that cell just ends up, water leaves, and it bursts, or it lyses, it shrivels, and that induces apoptosis in it. So kind of cool how this complement system forms this membrane attack complex. And then remember the difference between allergens and regular antigens. So allergens are different than antigens because allergens could they're considered harmless, but they produce an abnormally large immune response. Sorry, my English was off there. But produce an abnormally large immune response is something that should be harmless. And then understand the different immune cells. So, helper T cells, these are the main part of the immune system. If you don't have helper T cells, the whole immune system will fail. What a B cell is, what a memory cell is, what a cytotoxic T cell is, and how regulatory T cells kind of just monitor the whole system. So make sure you just, if they're used in words, matchings or something like that, you understand what the different cell types are. And then last thing here is the digestive system. So remember, the alimentary canal is also known as the gastrointestinal or GI tract. The process of swallowing is referred to as glutathion. The glottis, remember, is the opening, and then epi is upon the glottis. So when you swallow that food bolus, the glottis blocks the trachea. So if this was a trachea, it would block it so that that food bolus can move down the esophagus then. It's an amazing drawing there. And then know the main layers. So you have the serosa layer. So the whole canal has these main layers. So you have the serosa layer, and then, not the serosa layer, starting on the inside, mucosa layer, and then you have the submucosa layer, and then you have the muscularis layer. There's usually an externa and an interna, where one is running longitudinally and one is running circularly. And then you have the serosa layer on the outside. So serosa, muscularis, submucosa, and mucosa. And then these muscle layers, then remember, because one is circular and one is longitudinal, they move the contents of the GI tract via that peristaltic wave, or peristalsis. So then understand the anatomy of the digestive system too. This is a big one. So make sure you watch that digestive system lab video. This is the majority of the practicals based on this, and make sure you understand the lab models that we have, including that one flat picture thing. And then a little bit about the teeth here. Remember, hardest substance in the body is enamel. I'm not asking too many questions about the teeth, but one of the big ones would be that understanding what enamel is, has that covering on it, and it's very hard and protective. So, sometimes if you find something in the woods that's been there a long time, you can still find the teeth of that organism in some of the bones. And then here, so this is also kind of related to the immune system as well. So the most effective microbe-killing mechanism of digestion is actually the hydrochloric acid, or HCl, in the stomach. Most of the things we eat on our food are foods covered in bacteria. It's perfectly fine, but most of it gets killed in the small intestine. Not the small intestine, the stomach, by HCl. And the stomach is also unique. So I guess when in doubt on this exam, guess stomach, because it's involved in secretion, mechanical breakdown, digestion, and absorption. And it's also the only digestive organ with 3 muscle layers. Remember, it has the oblique, longitudinal, and circular, and that allows that mechanical breakdown of food a lot more. It also produces intrinsic factor. Remember, intrinsic factor is required for vitamin B12 absorption in the small intestine. And it also produces a mucoid barrier to help prevent self-digestion because that HCl would break it down too fast. And then when we have, a cell in the stomach, we have different cells that are found in it. So make sure you understand what each of these cells are producing, like parietal cells producing HCl, chief cells, enteroendocrine cells producing hormones, mucous neck cells producing an acidic mucus, and so forth. So make sure we review those as well. And then most absorption does take place in the small intestine. We're not absorbing nutrients in the large intestine. You can have some absorption, like from aspirin and alcohol in the stomach, so it's not no absorption. But most absorption happens in our small intestine because we have that massive surface area from all the villi and microvilli on top of them, and those microvilli contain those brush border enzymes which help break down carbohydrates and proteins as well. And then just some other parts here. The pancreas, it makes most of the enzymes for digestion. And remember, these are released in an inactive state. If the pancreas produce active proteases and lipases and things like that, it would eat itself. So it releases in an inactive state, something, like trypsinogen is then activated to trypsin and so forth. So it goes through this activation cascade to protect itself. Then also part of the anatomy, know the locations of the major sphincters that we have throughout the GI tract. So we have the cardiac sphincter coming into the stomach. Then you have the pyloric sphincter exiting the stomach. Then you have the ileocecal sphincter or valve, and then going through the large intestine and out. And again, knowing all the anatomy of going through the digestive system is important, mostly for the practical. I said this twice. So swallowing is referred to that. Oh yeah, I copied this and moved it up because I was talking about the mouth first. So ignore these last 2 lines here. I already said them. But that's a general overview of this exam. I thought this was a little easier going through it like this and being able to read it to you guys and then drawing all my red all over it. So I hope this helps. And then you can also pause this in parts. Again, I'm not sending this out as a Word file. Just continue reviewing and go through it and so forth. But you could take notes on this and so forth. But yeah, if you have questions on the exam, feel free to reach out to me. But if not, I wish you good luck. And I hope you have a great day. And bye-bye.